In this video, we will discuss about this effect of GalNAz and OST on the glycoRNAs. So far, we have discussed the results that shows that these glycoRNAs in the in the exosomes might help in the intercellular communication. So here they will check the effect of GalNAz on this glycoRNAs formation and also the effect of OST. OST is the enzyme that helps in the endoplasmic reticulum to transfer the sugar molecule precursor, that is uh, glucose 3 mannose 9 and glycnec 2 onto the NXST site of proteins. So if we inhibit that enzyme, whether it will affect the glycoRNAs, and also if we inhibit this, if we do some competitive reactions against this GalNAz, will that also affect the glycoRNAs or not? That we will check in this video. So before we going into the results, let us understand how this GalNAz work. So with this glucose 1-phosphate will undergoes the UDP glucose through this UTP conversions to PPI, and after that, with the help of this GAL reactions, it will undergo this UDP GalNAc, and this UDP GalNAc can undergo reversible reactions through UDP GalNAc to UDP glycnac using GALs, and after that, UDP glycnac can undergo MANAC 6-phosphate using GNE, and this MANAC 6-phosphate can either go to CMP sialic acid or it can leads to the GDP fucose reactions, GDP fucose formations. So if someone inhibits this GAL and GNE, then it will prevent this traditional GalNAc conversions to the CMP sialic acid or GDP fucose. So if they inhibit these enzymes and supplement the cells with the radioactive enzymes that we have discussed in the previous video, that is AC4 GalNAs, then it should increase the AC4 GalNAs conversion rate, because the native GalNAc conversions will goes down. So first they check the knockout efficiencies, and they knockout using this double stranded RNase 1, and they use 2 RNAs for each of this knockdown. It is for GAL knockdown they use 2 RNAs, and for GNE knockdown they use 2 RNAs, and we can see that the percentage of mRNA that are formed in this 72 hours of this knockdown is quite low, less than 50% for the GAL knockout, and also for the GNE knockout it is less than 50%. For one, it is almost equal to 50%. And then they check in these RNA the glycoRNAs formations. So once they confirm this knockout, what it is, they treat the cells with this double stranded RNAs. 24 hours after that, they inject this AC4 GalNAs, and after that, there's I think 28 or 48 hours after injecting of AC4 GalNAs, they check the glycoRNAs concentrations, and they are able to see that this glycoRNAs concentrations increases compared to the control, or compared to the control RNAs that they have used. And this indicates that it's the GalNAs that plays a role towards, or this GalNAc convergence pathway that is shown in the panel A, this plays a role towards, or has a role towards, these glycoRNAs formations. Then they wanted to check this OST formations, or when the glycosylation is completely stopped, whether it will also affect the glycoRNAs or there is some other pathways that is going on within the cells. So in order to do that, they inhibit this OST using the small molecular inhibitors NGI1, and then they check the cellular RNAs and exosomal RNAs concentrations at different concentration of this NGI1, and they can see that with increase in the NGI1 concentrations, they are able to see a decrease in the glycoRNAs. It can be seen for cellular RNAs and also for the exosomal RNAs, indicating that this OST, this glycoRNAs also follows this traditional method of this initiation of the glycosylation in the endoplasmic reticulum via OST. And these things, they have the penalties results are been quantified on the right hand side, where we can see that the glycoRNAs ratios goes down significantly compared to the control uh, for the cellular RNAs and also for the exosomal RNAs at different concentration of NGA1. So both of these results indicates that this GalNAz pathway plays a role towards these glycoRNAs, and also these OST pathways also plays a role towards these glycoRNAs formations. This is what they finally conclude in this paper. If you need to understand more about their experimental results and wants to check their supplemental results, you can go to the reference that has been provided in the description. So that's all for this video. Thank you for your time.